Hi, my name is Lance Capel. This is Grand Canyon University, and this class is Bio 520. I'm doing my cardiac presentation on the topic of electrical pacemaker activity of cardiac autorhythmic cells. The heart's rhythmic contractions are initiated and coordinated by specialized cells called autorhythmic cells. These cells have the unique ability to generate electrical impulses spontaneously, acting as the heart's natural pacemaker. And here's a breakdown of their key features. I have here the location, primarily found in the sinoatrial node, which is located in the right atrium. Small clusters are present in the atrioventricular node and specialized Purkinje fibers. Next, we're going to talk about the function of the autorhythmic cells. They generate spontaneous electrical impulses called action potentials. These impulses spread throughout the heart, causing coordinated contraction of muscle cells. The SA node sets the heart rate, again the pacemaker, due to its faster intrinsic rate compared to other autorhythmic cells. Now let's delve deeper into the intricate mechanisms behind this vital process. The heart's rhythmic contractions are a marvel of biological engineering, orchestrated by specialized cells called autorhythmic cells, which we talked about previously. These unique cells act as a heart's natural pacemaker, spontaneously generating electrical impulses that initiate and coordinate muscle contractions. So on this next slide, we have here the sinoatrial node, which we discussed earlier, the atrioventricular node, and the Purkinje fibers. Now, the sinoatrial node is located in the right atrium. The SA node contains a primary cluster of autorhythmic cells. These cells set the heart rate by generating impulses at the fastest intrinsic rate. The atrioventricular node, this cluster of autorhythmic cells, sits between the atria and the ventricles. It relays the electrical signal from the SA node, and if necessary, can take over as a backup pacemaker if the SA node fails. And then lastly, the Purkinje fibers. These specialized fibers conduct the electrical signal throughout the ventricles, ensuring synchronized contraction of the heart chambers. While not technically pacemakers, they play a crucial role in transmitting the impulse for coordinated pumping in the heart. And this next slide I have here, we're going to discuss the underlying mechanism. So unlike regular muscle cells that rely on external stimuli for contraction, autorhythmic cells have unique property. First, they have leak channels. These channels allow a slow, steady influx of sodium ions into the cell. This gradual depolarization, called the diastolic depolarization, builds up like a charging capacitor. Next is the threshold potential. As the sodium influx progresses, the membrane potential, which is the voltage inside the cell, gradually increases. Once it reaches a specific threshold, voltage-gated calcium channels open. Next is the calcium surge. The sudden influx of calcium 2 ions further depolarizes the cell, triggering an avalanche of further calcium entry, creating an action potential. The surge is like the release of stored energy, causing the cell to fire an electrical impulse. And then next we have repolarization. After the action potential, potassium channels open, allowing potassium ions to flow out of the cell, repolarizing the membrane and resetting the cell for the next cycle. Next, we're going to discuss the key factors influencing rate. So the autonomic nervous system, the sympathetic nervous system uses chemicals like norepinephrine to increase heart rate, while the parasympathetic system uses acetylcholine to decrease it. Next are the hormones. Stress hormones like adrenaline can also stimulate faster heart rate by influencing ion channels in autorhythmic cells. And then lastly, the physiological factors. So some physiological factors included are exercise, body temperature, and oxygen levels, which can all impact the rate of impulse generation by influencing the activity of ion channels and enzymes within these cells. And then next we're going to talk about the significance of this. So understanding this process is crucial for diagnosing and treating various heart rhythm disorders such as arrhythmias and bradycardia. Pacemaker devices mimic the electrical activity of autorhythmic cells to regulate heart rate artificially when natural pacemakers fails. And this next slide here, I have a little bit of explanation. So understanding the electrical pacemaker activity of autorhythmic cells is crucial for several reasons. Number one, diagnosing heart rhythm disorders, developing treatment options, understanding cardiac development and disease. So diagnosing heart rhythm disorders, irregularities in the firing pattern of autorhythmic cells can lead to arrhythmias like bradycardia, which is slow heart rate, or tachycardia, fast heart rate. Analyzing the electrical activity can help identify the causes of these arrhythmias. Developing treatment options. Pacemaker devices are designed to mimic the electrical activity of autorhythmic cells, artificially regulating heart rate when the natural pacemakers fails. Understanding the mechanisms involved helps design and improve these devices. And understanding cardiac development and disease. So studying the development and function of autorhythmic cells can shed some light on congenital heart defects and impact of various diseases on cardiac rhythm. Next is the heartbeats mastermind. So I kind of pulled everything together here and made a little chart. So again, just to review, the location of the autorhythmic cells, locate specialized cells in the SA node, the right atrium and other locations. Again, the function, they generate electrical impulses, action potentials that trigger heart contractions. The mechanism, unique leak channels allow gradual depolarization, triggering calcium influx and action potential. And then pacemaker, the SA node sets the rhythm due to its faster intrinsic rate. And here are some more things I added. Rate influencers, such as the autonomic nervous system, hormones, and physiological factors. The significance, it's crucial for understanding and treating heart rhythm disorders. And then I believe there should be, further exploration. Action potential, cell types, and clinical impact can be explored further down the line. And these are my references. and I hope you've enjoyed my presentation. Thank you.